Hello everyone, today we will present our project on pneumonia detection using chest x-rays. Our study focused on a hybrid CNN-VIT architecture and CAI analysis using GradChem++ and SHAP. First, we will introduce the project overview and motivation. Then we will explain the dataset, model architecture, and experiments. Finally, we will present the results, XAI analysis, additional study, and conclusion. First, this is the project overview. This project is a deep learning system that classifies pneumonia from chest X-ray images. We used a hybrid structure combining CNN and VIT along with GradChem++ and SHAP-based XAI analysis. Next is the motivation of our project. Recently, increasing numbers of imaging examinations have raised radiology workload. In addition, chest X-ray interpretation can vary depending on reader experience and fatigue. Another issue is medical accessibility. In developing countries or regions with limited medical infrastructure, radiological interpretation itself may be difficult. Therefore, we focus on the potential of AI-assisted systems as medical screening tools. Next is the dataset and pre-processing. We use the Kermany pediatric chest X-ray dataset. The dataset contains about 5863 images, including 5216 training images and 624 test images, classified into normal and pneumonia classes. Before training, we applied image resizing, augmentation, and normalization. Next is the model architecture. Our model uses a parallel branch hybrid structure with CNN and VIT branches. The CNN branch uses 6 convolution layers to extract local features such as opacity and texture patterns. The VIT branch divides the image into 10 by 10 225 patches and learns global relationships using self-attention. The extracted features are fused and passed through fully connected layers for final classification. This slide explains why we use the hybrid structure. CNN is effective for local lesion detection but limited in capturing global long structure. In contrast, VIT is strong for global context learning but less stable on small medical datasets. Therefore, we combine both approaches to achieve more balanced classification performance. Next is the training setup. We use the Atom optimizer with a learning rate of 0.0005. Binary cross entropy was used as the loss function. We also applied early stopping to stabilize training. Next is the XAI design. In medical AI, explainability is very important. We use GradChem++ and SHAP for XAI analysis. GradChem++ visualizes attention regions using heat maps, while SHAP explains feature contribution. Next are the baseline results. The CNN-only model showed relatively stable performance. However, the VIT-only model failed to properly classify normal images, showing almost zero recall for the normal class. This result demonstrated the necessity of the hybrid structure. Next are the results of the proposed model. The final accuracy was about 91%, and pneumonia recall reached approximately 0.9744. Since missing pneumonia cases is critical in screening, this high recall is especially meaningful. These characteristics are also confirmed in the confusion matrix. Out of 390 pneumonia images, 380 were correctly detected, with only 10 false negatives. This indicates that the model missed very few pneumonia cases, which is important for screening applications. This slide explains how we interpreted the GradChem++ and SHAP results. GradChem++ visualizes which regions the model focused on during prediction, and brighter areas indicate stronger activation. SHAP shows how each region contributed to the prediction score. In SHAP visualization, red regions increase the probability of pneumonia, while blue regions contribute to our normal prediction. This is a correctly classified normal sample. The GradChem++ heat map mainly focus on long regions. In the SHAP result, blue regions contributed to our normal prediction. This suggests that the model correctly focused on meaningful normal long features. This is a correctly classified pneumonia sample. GradCam++ highlighted opacity and infiltration-related regions. In SHAP visualization, red regions contributed toward pneumonia prediction. This suggests that the model focused on abnormal long patterns during prediction. To further validate the effectiveness of the parallel hybrid approach, we explored a sequential CNN to transformer structure. Unlike the parallel approach, this model sequentially refines CNN features through a transformer encoder. In this structure, CNN first extracts feature maps. These features are converted into tokens and passed into the transformer encoder for contextual refinement. The final classifier then predicts either normal or pneumonia. For the additional experiment, we adjusted hyperparameters such as learning rate, batch size, dropout to stabilize training. The sequential hybrid model achieved about 90.87 accuracy and showed relatively balanced classification performance. In addition, the model produced 36 false negatives during pneumonia detection. Next is the comparison between sequential and parallel structures. The sequential model showed more balanced recall, while the parallel model achieved higher pneumonia recall and fewer false negatives. Finally, we selected the parallel hybrid structure. In medical screening, high sensitivity and low false negatives are more important than overall accuracy. Finally, this slide summarizes the limitations and significance of our study. This study was limited to a single chest X-ray dataset and binary classification. In addition, XAI results should be interpreted as supportive evidence, not direct medical diagnosis. Despite these limitations, the parallel hybrid model showed stronger screening-oriented performance with higher pneumonia recall and fewer false negatives. Thank you for listening.